Poliomyelitis or polio. Poliomyelitis is an acute infectious disease that affects the central nervous system CNS when it occurs in serious form. The flaccid paralysis resulted by the destruction of motor neurons in the spinal cord. The most poliovirus infections are subclinical. Structural properties and composition of the poliovirus. It consists of a capsid of 60 subunits, each of 4 proteins VP1, VP4. Arranged in icosahedral symmetry, the genome is made up of a single-stranded positive sense RNA, constituting 7.4 kilobits. It is polyadenylated at the 3' end and has a VPG protein covalently bound to the 5' end. The RNA genome is considered infectious because it is a positive sense RNA, which can directly serve as mRNA upon entry in the host cell. Sensitivity and stability of poliovirus. They are inactivated at 55 degrees Celsius for 30 minutes and by 0.1 ppm chlorine. Much higher concentrations of chlorine are needed to disinfect sewage containing viruses. It is not affected by ether or sodium deoxycholate. Antigens of poliovirus. There are 3 antigenic types of polioviruses based on epitopes found in the VP1, VP2, and VP3 proteins. Replication or life cycle of poliovirus. The replication starts in the cytoplasm of the host cell. The virus attaches to the particular receptor in the cell membrane. Receptor binding triggers a conformational change in the virion, i.e., the loss of VP4, which results in release of the viral RNA into the cell cytoplasm. VPG is removed from the viral RNA as it associates with the host ribosomes and translation starts. Replication and life cycle of poliovirus. The viral RNA is translated into a polyprotein that contains both capsid proteins and essential replication proteins. This polyprotein is rapidly cleaved into fragments by proteinases encoded in the polyprotein. Replication or life cycle of the poliovirus. Synthesis of new viral RNA cannot begin until the virus-coded replication proteins, including an RNA-dependent RNA polymerase, are produced. The infecting viral RNA strand is copied, and that complementary strand serves as template for the synthesis of new plus strands. Many plus strands are generated from each minus strand template. Replication and life cycle of poliovirus. Some new plus strands are recycled as templates to amplify the pool of progeny RNA. Many plus strands get packaged into virions, the protomers. VPO, VP3, and VP1 assemble into pentamers that package plus-stranded VPGRNE to form provirions. The provirions become infectious when a final cleavage changes VPO to VP4 and VP2. The mature virus particles are released when the host cell lyses. The whole replication cycle takes 5 to 10 hours. Pathogenesis of polio. The mouth is the portal of entry of the virus, and first multiplication occurs in the oropharynx, or intestine. The virus is present in the throat and in the stools before the onset of illness. After 1 week of infection, there is low virus in the throat, but it is excreted in the stools for several weeks and is present in the blood. The virus first replicated in the tonsils, the lymph nodes of the neck, Peyer's patches, and the small intestine. Pathogenesis of polio. The CNS may then be invaded through circulating blood. Poliovirus can spread along axons of peripheral nerves to the CNS. It continues to move along the fibers of lower motor neurons to involve the spinal cord. Poliovirus invades certain types of nerve cells and its replication may damage or completely destroy these cells. Pathogenesis of polio. Poliovirus does not multiply in muscle in vivo. The changes in peripheral nerves and voluntary muscles are resulted by the destruction of nerve cells. Some cells that lose their function may recover completely. Inflammation occurs due to the attack on the nerve cells. Clinical manifestations and symptomatology. The response to virus ranges from an apparent infection without symptoms to a mild febrile illness to severe and permanent paralysis. Most infections are subclinical, but only 1% of infections result in clinical illness. The incubation period is usually 7 to 14 days, but it may range from 3 to 35 days. Clinical manifestations and symptomatology. 1. Mild disease or prodromal phase. The patient has only a minor illness, manifested as fever, malaise, drowsiness, headache, nausea, vomiting, constipation, and sore throat. Recovery occurs in a few days. 2. Non-paralytic poliomyelitis or aseptic meningitis. In addition to the symptomatology of the mild phase, the patient may experience stiffness and pain in the back and neck, which may persist for 2 to 10 days, and recovery is rapid and complete. Poliovirus is only one of many viruses that produce aseptic meningitis. Rarely the disease progresses to paralysis. 3. Paralytic poliomyelitis. The flaccid paralysis results from lower motor neuron damage, non-coordination secondary to brainstem invasion, and painful spasms of non-paralyzed muscles may also occur. The amount of damage varies greatly. Maximum recovery usually occurs within 6 months, with residual paralysis lasting much longer. 4. Progressive post-poliomyelitis muscle atrophy. Recurrence of paralysis and muscle wasting has been seen in patients decades after paralytic poliomyelitis. The progressive post-poliomyelitis muscle atrophy is rare. It is resulted due to physiologic and aging changes in paralytic patients already burdened by loss of neuromuscular functions. Laboratory diagnosis. The virus may be isolated from throat swabs, rectal swabs, or stool samples. Tissue cultures of human or monkey cells are inoculated, incubated, and observed for cytopathogenic effects. An isolated virus is identified by neutralization with specific anti-serum. Virus can also be identified more rapidly by polymerase chain reaction PCR. Serum specimens may be used to determine antibody titer. Prevention and control. Both live virus and killed virus vaccines are available. Formalin inactivated vaccine stock is prepared from virus grown in monkey kidney cultures. Killed virus vaccine induces humoral antibodies but does not induce local intestinal immunity, so that virus is still able to multiply. In the gut, live attenuated vaccine sabin is grown in primary monkey or human diploid cell cultures and delivered orally. Control and prevention. The live polio vaccine infects, multiplies, and immunizes the host against virulent strains. In the process, infectious progeny of the vaccine virus are disseminated in the community. The vaccine produces not only immunoglobulin IgM and IgA antibodies in the blood, but also secretory IgA antibodies in the intestine, enabling mucosal immunity. Control and prevention. Both killed virus and live virus vaccines induce antibodies and protect the CNS from subsequent invasion by wild virus. However, the gut develops a far greater degree of resistance after administration of live virus vaccine.